Sir, please go ahead, sir.
Okay, thank you, one and all. I am here to present COMFA and COMSIA, a 3D QSR techniques. They are decades old, but still they find niche in the drug discovery and drug design. Okay, they are still promising in the drug discovery and drug design. Okay, so I believe that it may find your interest. They are very important techniques actually in the field of drug design, computer-aided drug design techniques, especially take into the consideration. Okay, so the history of drug discovery, my dear colleagues, my dear friends. So, if you just go back to the history of this drug discovery over a period of time now, so early 19th century, if you take into consideration, in the early 19th century, that means 2 centuries back. Okay, so at this, in the early 19th century, the situation was such that it was more often natural products which were there in the market. For example, like morphine, cocaine. So such natural products were there in the market. Okay, so as the time proceeded, okay, if you come to the late 19th century, when you moved from early 19th century to the late 19th century, there was a drastic change that there were only fewer natural products were there in the system of allopathic system of medicine, and it was more often synthetic substances were there in the pharmaceutical market, in the market, like that. So at that time, what really has happened is that, so fewer national, at that time, what really this pendulum shift which has been taken place, why it has happened, the reason is that many of these dye and chemical making companies, okay, they started doing the pharmaceutical research. One such example is the Bayers. Okay, Bayer is the company today, is a well established pharmaceutical company, but it was basically, once upon a time, it was a dye and chemical manufacturing company. Okay, at that time, so if you take regulations for the dye and chemical companies and the pharmaceutical companies, lot of difference will be there. Okay, but at that time, in the late 19th century, in the 19th century, so there was an option for even chemical companies or dye making companies also to bounce back to the pharmaceutical company, okay, to make their profits in selling the drugs, like that. But when we moved from 19th century to the 20th century, okay, in the just in the beginning of 20th century, one scientist makes it very clear, that is John Langley. He makes it very clear that the theory of receptive substances, in the sense that the concept of binding a drug to the receptor. Okay, so drugs will go and bind to the specific receptors in our body. So that concept, it makes it very very clear at the beginning of the 20th century. Okay, so then what happens is, so drugs thereby initiating the biological effects or inhibiting the cellular functions. Okay, so this is all attributed to the, because of the drugs which will bind to the receptors. Okay, like that. So next, what has happened is, in the early 20th century, then the people, especially the scientists across the world, they started working on the concept of rational drug design. Okay, this term rational is, for the first time it was been used in the 1909. That means something, it is conceptual, hypothesis based kind of a drug design. Okay, so till that time, there was no concept of rational drug design kind of a concept, like that. So the goal of this rational drug design was the safer treatment, to give a safe treatment. That was the goal at that particular point of time. So here two scientists, they come forward. One is Paul Ehrlich and Zachariah Atta. So these Paul Ehrlich and Zachary Atta, they come forward to maximize the therapeutic index. As we all know, what is the therapeutic index? The amount which causes effect to that amount which causes the toxicity. That needs to be enhanced in the form of a therapeutic index. So in that context, they synthesize some large number of compounds. That means that they synthesize some 600 compounds. They synthesize these molecules and evaluated their ratio of minimum curative dose to cure the disease and maximum tolerated dose. Okay, so in that way, by doing this to find out the therapeutic index, and they found salvarsan. Okay, so this salvarsan, okay, this drug they found by screening 600 molecules after synthesizing, analyzing them, confirming the structures, and then screening, okay, to find out the maximum curative dose to, and maximum tolerated dose to find out the therapeutic index. They find this salvarsan. You find, just look at this structure of salvarsan, my dear friends. So it has, you can see that there are two arsenic atoms are there. Okay, today if you do this limit test for arsenic, it will never passes the limit test for the arsenic. Okay, okay, but it was being used as a drug at that point of time. In the past we have used it as a drug. Okay, it was very unfortunate thing that such kind of an structure, such kind of an molecules as a drug, we have used it as a drug. Okay, so then, as you all know that this salvarsan, which has been used as an anti-infective at that particular point of time, then it was been replaced with the penicillin in the year 1940. So Alexander Fleming discussed penicillin, and that replaces this salvarsan in the year 1940, like that. So my dear colleagues, why I'm stressing on all these things is that this is what has happened in the past. We should know that what has happened in the past, and we should, in that background, we should think what next to be done in the future, present, and the future, like that. Okay, so in this context now, in the year 1960s, that means in the, from early 20th century to the late 20th century, in that time the first successful attempt to relate the chemical structure to the biological action was quantitatively done by the Hans in the form of an QSR, quantitative structure activity relationships. Okay, so 1960, there the Hans comes forward and comes out with the term, that is QSAR, for the first time. Okay, so everyone get fascinated with this QSAR, quantitative structure activity relationships. We'll see about, because this COMFA and COMSIA techniques are basically and QSAR based techniques, we'll see much more detail about them later. Okay, but before that, let us understand the background, in what context this QSAR or COMFA COMSIA evolves, and in that context, if we study what is QSAR, what is COMFA COMSIA, what kind of techniques they are, what is the protocols, how we can find the results, all these things we can better understand. Okay, so mid to the late 20th century, what has happened is that understanding disease states, biological structures, okay, proteins, their functions, their process, distribution, transport, distribution, and metabolism. So medicinal chemists use this knowledge to modify the chemical structures to influence the drug's activity. Okay, with the advancement in the computers. Okay, so with the advancement in the computers, people started using, scientists across the world, how we can advance it. Say, my dear colleagues, my dear friends, the much of the development in the field of pharmaceutical sciences happened in this particular time. Okay, from mid to the late 20th century. today if you look at the in the medical shop, what are the drugs that we can find it in the medical shop, most of these drugs were being discovered in the from mid to the late 20th century. Okay, the majority of the drugs that we are using it, that we are taking today, they are this uh, from this mid to the late 20th century, like this. Okay, so now the drug discovery and drug development, as we all know that the time from the conception of an approval of to the approval of a new drug, it takes about 10 to 15 years of time. It takes such a long time, but still, if a company can able to achieve it uh, at a lesser time, it is really benefits that particular company. Okay, and the cost to bring to the money it will takes. Okay, so the cost increases every year, so maybe some 10% of the cost it may increase every year because of the inflations. Okay, so what in this context, what I mean to say is that sometimes what happens is that in pharmaceutical company, after spending some 10 to 15 years of time, even then they could not able to succeed. Okay, they cannot succeed even 10 years or 15 years of doing the research like that. So the in this context, what happens is that a company which does the research in pharma field as part of the research and development, it should have strong financial backup. So otherwise what happens is, after spending sometimes so much of time, they still, it may be a failure. Okay, they cannot come out with a drug to the market. So such failures also it should be able to sustain. That is not that easy. So that is the reason why most of these um, drug research based companies, they are basically multinational companies, and very indigenous within our own country. Very few companies are coming forward to do the research and development to develop the drug to the market, and like that. And in a country like in our country like India, we are still struggling to find a drug and release that drug to the market. Okay, so very few success we have, still a long way to go in meeting the standards of global standards, so that we can able to bring the drugs to the market like that, in the global market like that. So how our drugs are discovered? Okay, so this is a big question that okay, one traditional way is classical way of drug discovery. But what happens is that many of you, believe it or not, many of the drugs which are there now in the medical shop, which are there in the market today, they are mainly discovered by an accident. Why it happens so like that? That means that whatever we have understood, the physiology, the biology, and in that context, the drug discovery, if we take into consideration, our understanding is not that good. It means to say that, so that because of that, what happens is this drug discovery, somewhat marginally, it becomes like an accident-based drug discovery, like that. Majority of the drug discoveries, if we take in consideration, okay, even though your intentions are different, even if your intentions are also the same, but still what you are anticipating in that results of during the process of drug discovery, they are not aligned with what is the real drug which comes ultimately out of that research like that. So there will be some element of some kind of an accident, serendipity will play a very important role in discovering the drugs like that. So a keen observation is very important for the drug researchers with a keen observation, one can able to find out what may be the reason. Even though we are expecting one kind of a result, but we won't get it in that way. Some molecules on the top may get failed, and some molecules in the middle they may get succeeded because of various reasons. Okay, so one really cannot expect at that point of time like that. So mainly it is mainly because of serendipity. Okay, the fact of the matter is that today we are discussing majority of the drugs what we have discovered, they are mainly by the accident like that. Okay, so maybe with better understanding of the biology, chemistry, all the different disciplines of the uh, drug discovery if we take in consideration, so we may able to discover the drug as we wish like that. Okay, hope so in the near future we do it in that way. So but you can see that drugs can be discovered mainly by screening of the new drugs. Okay, instead of saying screening of new drugs, we can say screening of new ligands, we can say it as. So ligands that bind to receptors, so these ligands can be of diverse libraries. We can synthesize and we can keep it as a libraries of compounds, just like how in your own college you'll find a library, where in that library you'll find the different books. So in that way, we can find that some kind of libraries of compounds. And so using these libraries of compounds, we can do the high throughput screen. Yogesh, sometime. So we can do the high throughput screening. So and in the pharmaceutical industry scenario, if we take into consideration, we do the high throughput screening. Okay, in the sense that large number of compounds. Okay, we can screen it in a the scaffolds, the nucleus should be varied in these class of ligands. Okay, so that we can screen it with the diverse structure of these ligands, and we can find the hits which are ideal to be developed as a lead compound for that particular case of an drug research take into the consideration. Okay, so that is one way of doing it. That is what the pharmaceutical industries are doing it right now, and some of the academics also will do it if they provided, if they have a good infrastructure, good facility, they do this high throughput screening. But nevertheless, uh, what happens is most of the academics, what we do it is just we try to design the molecules, we try to synthesize them, and we try to analyze these compounds for their structures, and then we'll screen it for their activity. Pharmacological screening we do it. That's what we normally do it. Okay, little extension of it is the high throughput screening with large number of compounds, and developed assays to do this kind of screenings, pharmacological assays for biochemical uh, estimation, for drug binding to the receptor, and also for functional assays in the cellular functions. So then we can take it to the pharmacological assays on the animals like that. It is all part of the preclinical evaluation of the drugs. So the other way of in the drug discovery is that modification for the improvement. In the sense, it is just like, for example, any new car, if it comes to the market, uh, naturally everyone will look at that particular car, and if that car selling is more, if the sales of that particular car is more, if more and more people are liking that particular car, then what happens is they naturally get look at that particular car, other competitors, and they try to modify that car so that they can come out with a newer car which is better compared to the earlier car which has come to the market like this. So in that context, the modification for the improvement will be there. The room for the improvement will be there. Okay. In this case, we come into two terms here. One is first in class and the best in class, like that. Okay. So first in class means amongst the class of that particular compound. Okay. So what is the first compound that can come to the market? For example, if you take penicillin. So amongst the penicillins or beta-lactams, if you take in consideration, penicillin is the beginning part. That is the first in class. It did wonder. It was there in the market for a longer time. It was exceptionally well. Okay, but today, if you take in consideration, the pharmaceutical companies look at that first in class structure, its function. So they try to modify that structure and try to bring out the newer drug into the market, which is better than the existing drug, like that. So the best in class will be an everyone will be hunting for the best in class. Okay, so that is very important to make the money in the pharmaceutical market, like that. So another important approach is mechanism based drug design, by with the understanding of how this drug is going and binding to the target receptor or enzymes. Okay, so that mechanism of this drug, if we take into consideration, so based on that we are going to develop the we are going to do the designing of the drugs. So that is like that. Sometimes what happens is we use combination of these techniques. We will be using it different combinations, we use it in different ways. So that is by combining the techniques is the one such technique, by combining the techniques. Okay, so this is how we discover the drugs like this in the very um, broadly, if I take into consideration. Okay, now this is how the drug discovery goes. Everyone knows that first we identify disease on which we have to do the drug research. So that disease is a manifestation based on the symptoms. The symptoms are in turn connected with the biochemical pathways. Okay, because these biochemical pathways will get disturbed. So due to maybe because of accumulation of more of the metabolites or less of the metabolites, you find some kind of a disturbance in the state. So that will be manifested in the form of a disease with the symptoms. So if you identify that biochemical pathway, again if you go deeper into the biochemical pathway, there will be different enzymes will be taking part. Okay, so there which enzyme is malfunctioning, so we have to understand that, and we have to identify that particular enzyme, which is basically a protein. Sometimes it may be an enzyme or sometimes it may be a receptor. Then we should try to find a way to produce this protein by recombinant DNA technology, so that we can find it abundant amount, so that we can do the screening of libraries of compounds. Okay, as we say, if you want to screen the libraries of compounds or the ligands, then you should have a protein, so that you can do the ligand protein binding studies, and with that we can come out with certain ligands which are so then what happens is we'll we'll then the preclinical testing. So with the so with the help of medicinal chemistry and pharmacology, so the next stage it goes to the evaluation, and then filing an investigation new drug, and parallelly the formulation will starts, and then the uh, clinical trials will starts, and then filing in new drug applications, and then getting an approval of the FDA so that the pharmaceutical companies can sell the drugs globally like that. So this is how the uh, process of drug discovery will be happening. Now, in this context, what we will be looking into the consideration is the early part of the drug discovery taken into the consideration. Okay? So, in this early part of the drug discovery, the role of medicinal chemist will play a very vital role. Okay? So, we are basically in chemists. So, we are in medicinal chemists. We are in the pharmacy field. If you are in chemistry, pharmaceutical chemistry. Okay. Of course. Uh, to some of the context, since it is drug discovery is multidisciplinary, you cannot restrict only to the chemistry like that. You are a medicinal chemist, you should have a sound knowledge of design also, synthesis also, analysis also, and also evaluation also. You must have a fair amount of knowledge in all these areas of the drug discovery. So next, so in this early part of the drug discovery, what we look at is the protein and the ligand. So we need to synthesize the ligands, and we need to check its binding affinity to the receptor or an enzyme protein structure. Okay, so for that we'll be doing either by high throughput screening. We use we screen hundreds, thousands of lakhs of molecules per day, and we get a trend of which molecule is hitting the target, which is not like that. So we'll get an so with that we can find out the hits, and with the hits we can compile it and come up with the lead structure, and with the lead optimization will be there. So we need to optimize the lead uh, based on the analog design. So so everywhere the drug design comes into the picture here. Once the ligand comes into the picture and the protein comes into the picture, the drug design will starts. Okay. So the combinatorial chemistry is another way of where we can produce large number of compounds in quick time. Okay. So and molecular modeling. Okay, where we can use computer graphics and computer models to improve the activity like that. So in vitro and in silico ADME models are also being used in this context, so that we can able to find out the efficacy of the molecules in the in vitro models as well as and compare it with respect to the in silico models in terms of their efficacy, and the ADME models take into consideration. So this is a part of the preclinical testing. So animals will be used for that. So animal ethical committee clearance will be the important aspect. So one has to clear that hurdle and process for the animal studies. Like so now, if it come to the our area of interest now for today's topic, if we take into consideration with related to the computer-aided drug design, this whole workshop of this in silico drug design on this refresher course is focused on. So now in this background of drug discovery process, now we are going to concentrate on the computer-aided drug design. The computer-aided drug design, mainly there are two ways of doing the computer-aided drug design. One is structure-based drug design. Based on the structure of the protein structure, so we are going to design the drug like that. Okay. Suppose if you have a lock and key, if you have lost the key for that particular lock, then what we can do is that by looking at that lock, we can always design the key for that, and we can open that key. So such is the concept of structure-based drug design. By looking at the structure of the protein, we are going to design the ligand that can able to fit into that particular protein structure like that. Next is ligand-based drug design techniques. Sometimes what happens is that you may not have a complete awareness of the protein structure, or even if you have, you may believe in the ligand-based technique, in the sense you have some of the molecules which are already going and binding that to that protein and exerting the desired pharmacological action like that. Okay, so that means that so in that case what we do is a ligand based drug design, we can use it. It is purely if you have some uh, just like if you have lost the key and if you go to the key maker comes with bunch of keys, and with those bunch of keys he tries to design one key that can fit into that particular lock like that. Okay, so it is just like that. So using some ligands into the consideration, by looking at the ligands, so we are going to design the ligand that can able to fit into that particular protein structure, but also take into consideration of its admit properties also fits in, its pharmacokinetic properties fits in, and its pharmacodynamic properties also fits in. Okay, so this is very important. And a drug is something that it is the right balance of pharmacokinetic and pharmacodynamic parameters. Both should balance it in such a way that for that particular case, for that particular disease, in that particular case, if you take in consideration, it will be a drug. Okay, whatever the formulation you do it, that is a different thing. So now this is what we do it as part of the computer aided drug design, either by structure based drug design or either by the ligand based drug design. You must remember these two broad techniques of a computer aided drug design. So this is now in this context, everyone says that my own colleagues, many of your colleagues may tell that molecular modeling. Like, what is this molecular modeling, my dear friends? So they say molecular modeling. People say I am doing molecular modeling. I want to learn molecular modeling like that. What is modeling? Okay, so it is just simple that, for example, in your B Farm chemistry labs, for example, if you are teaching in B Farm graduate students or an M Farm graduate students, if you are teaching them, while teaching them, because these tiny small ligands chemicals we cannot see them by our naked eyes, so what we can do, we try to build these stereo models, for example like this. Okay, we build in stereo models and try to understand how these molecules are existing in this nature like that. Okay, so but this is not the volume size shape. Okay, it is bigger. Okay, but this is how it exists at the, okay, at its own size levels if we take into consideration. So what is meant by molecular modeling? The 3D representation of the molecules based on the graphic or mathematical representation of the chemical structures. So in the sense that, that means how these molecules exist in this nature, like that we are going to simulate them the way how they exist, like just like for example how we are existing in this nature with this shape, size, volume. So just like that, these tiny small chemicals also exist in this nature in a similar way, like, but we, okay, so what happens is when we are trying to simulate these molecules on the computer screen, the question is that, the big question is that for the computational chemists is that how accurate these representations are. Okay, that means that's how they exist in this nature. Are you sure about that? This is a big question. Okay, so it is because why I'm telling is that, in what context I'm telling is that while you are modeling it, okay, in the graphics, that means in the silicon graphics or in the computer drug design, any ligand you sketch it first. After sketching it, you will minimize its energy and you do the conformational analysis to find out the right conformation for that molecule. Now, how to assess that it is the right conformation, right structure of that three-dimensional structure of that molecule? That is a big question. So whether it may be in protein, protein is still complex molecule, very difficult to attain the real structure, how it exists in this physiological environment in the body, and what we are trying to simulate in the computational way in the using the computers like that. So my assumption is that if we can able to predict the structure of the ligand exactly how it exists in this nature, and if we can have the structure of the protein exactly it exists in this physiological environment, then only we will get a successful results. So this is a big challenge. Okay, so whatever the softwares that we have today in the pharmaceutical market, they are all trying to match up with this concept. Now how well we can predict it, the ligand structures and the protein structures, so that the results of in silico matches with the wet lab experiments results. That means protein binding studies, wet lab results, okay, functional assays, they get matched with them. So this is a big question, and this is what we have to learn a lot, especially in development of the any algorithms for the any software development uh, packages like that. So the qualitative versus quantitative models, they relate the structures of conformation to the energy versus the mathematical equations. So that is what is molecular modeling. So altogether, if I want to say, if you are finding it difficult to understand what is meant by molecular modeling, it is very simple that trying to simulate the molecules the way how they exist in this nature. Okay, that's all we all try to do as part of the molecular modeling. It is as simple as that. Okay, so next, now coming back to the introduction to the CAD, now computer-aided drug design take into the consideration now. So in the computer-aided drug design, okay, this computer-aided drug design lies in the hands of an computational scientist. Okay, so now the thing is, how about these hands off? Now, if it lies in the hands of a computational chemist or scientist, suppose if you think that you are a computational scientist like that, now the computer-aided drug design, what you do, what you report, it lies on you. Okay, in the sense that what you are understanding about that particular concept and how you are going to adapt that particular concept to, okay, in that particular case of drug research take into the consideration, so that you can effectively use it, okay, for the drug discovery like that. Rather, it is a very complex process involving many scientists working from the various different streams like that. Say, remember my dear colleagues, that Eh, now you all know that some computational softwares are available. For example, Schrodinger is there. For example, Discord Studio is there. For example, MOE is there. For example, StarDrop is there. For example, okay. So, for example, Sibyl is there. Okay. So many software packages are there in the market now. Right? Okay. Now, how they are developing this software packages, what all the people involved in this development of the software people, software development, the software package development. So if you assess that, there are scientists who are working from different disciplines, they work together and develop the software package. From which different disciplines they'll come from? They have to come from physics, because your energy minimization molecular modeling is also physics dependent. They should be a chemist, because these structures are basically chemicals, they are chemical structures. Okay, and you need a biologist, because when we handle these protein structures, it should be a biologist, so they know better about the biologicals. Molecular biologists, they know about molecular biology of the molecules. Next, so in order to manipulate these molecules on the screen, and also sometimes we'll also use information technologies, the computers, the processing, the so and algorithms writing for that one. So programming languages we take into considerations. So next is the mathematics also. We use plain mathematics also for data, for any if you want to do any docking, or if you want to do any QSRs, we use the um, data, and it may be in plain mathematics, or it may be sometimes it may be in statistics. Okay, also we need statisticians in the team. So like that, from scientists from different disciplines, they gather together, find out, and their frequency should match, and they should their because there are plenty of ways of developing the softwares. So there everyone, if they think it, conceive one particular idea, and for that they will be developing the software package like that. So it's all depends, the success will all depends on the, in that particular team of scientists who are developing that software package, how well coordinated they are, how much understanding they have themselves, so that they can come out with the better software packaged like that. So don't you think that it is not that easy? Yeah, okay. So finding many people and many different ways of thinking, they have to come to some kind of different platform where they can able to design the software package for that. So that is a big task which is there amongst the everyone like that. So rather, it is a very complex process involving many scientists. Okay, because of this reason. Like next, now in the in silico drug design and virtual screening, there are four quadrants which we follow. Okay, so in the in silico drug design and virtual screening, there are four scenarios are there. What are these four scenarios means? Okay. So in terms of ligands and in terms of protein structure, whether you know about the protein structure and whether you don't know about the protein structure, whether you know about the ligand structure and whether you don't know about the ligand structure. Based on that, there are four quadrants of the drug design, computative drug design. So you must know these quadrants very clearly. Okay. So amongst these four quadrants, one is that if the known, if the known protein structure is there and known ligands are there. This is the first quadrant. Okay, that means protein structure is also known and ligand structure is also known. So then what we do is we do the structure based drug design technique, since you know about the structure is known. Okay, so structure of the protein is, if it is known, that means log information you have to design the key. So that is a very straightforward case and very easiest case in the computational drug design. If you have both the information about the structure of the protein as well as structure of the ligand. So protein ligand docking we can do the simulations, we can do, okay, like this. So in every case of this computational drug design, okay, the artificial intelligence also will comes into the picture. That is it, today's forefront of it like that. So now if the no known ligand is there but the known protein structure is there, we can do the de novo drug design. De novo drug design we can do it. Okay, so how do we do this de novo drug design? That means by looking at the structure of the protein, we build the structure of the ligand, okay, which is complementary to that particular protein. Okay, the protein is the target structure, and by looking at the target protein structure, we are going to design the ligand. So that is de novo drug design. Now in case like when there is known ligands are there but the protein structure is unknown, then also lot many things can be done with the known ligand structures. Okay, so that means what is quadrant comes here, this third quadrant, this can come to the first quadrant also, because in the first quadrant the ligands are also known. Okay, it not necessarily the third, first and third are interlinked. Okay, because They both are with the case of a known ligands. So now in the ligand-based drug design, what happens is one or more ligands are being required at least, so that we can do the similarity searching. Okay, so that means that if you have an one potent molecule, okay, so then we can search that potent molecule against the libraries of compounds and search the pharmacophore features which are matching against the library of compounds. And if you have several ligands also, what we can do is we can generate the pharmacophore out of that, and then we can use it virtually. We can screen the library of compounds. Like then what we can do is we can do the quantitative structure activity relationships. We can do QSAR. We can do it. Okay. So what we do it in quantitative structure activity relationships is all about what we are going to discuss in this class now. Okay. So next is drug likeliness screening. So your Lipin's case rule of 5 is also based on ligand based. Okay, even though it is connected to the admin properties. Okay, so next is in case if you don't have no ligand or no protein structure, usually normally in normal circumstance CAD is of no use, because CAD needs some kind of an input data. If you need some kind of an input data so that you can come out with an output data, so some kind of an input data is being required for this uh, CAD line. But still, sometimes what happens is, if for example, if you take high throughput screen line, okay, so if you do an in vitro biochemical estimation by biochemical assays in a high throughput screening way, and if you get a data of that hits which I'm getting, if you want to analyze it again, that can be used it in this quadrant of like this, in the fourth quadrant like that. But still it also expects some kind of data from the high throughput screening data like that. Okay, so that is why we say that in generally CAD is of no use if you don't have any data. We need some kind of data to analyze, and we can build some hypothesis based on the particular data like that. Okay, so next is now in this context, you see, it is difficult like this. You can see in this figure, like in this drug discovery, finding a drug is finding a needle in the haystack. How difficult it is that to find a needle in this haystack? Okay, if the needle becomes very bigger and bigger, it is easier and easier to do the drug discovery. So as part of the drug design, if you make the protocols as simpler as is possible and as predictive as it can be possible, so then the drug discovery will becomes easier and easier if you take into consideration. So in this context, the QSR comes into the picture in this, as we have seen it in the third quadrant, when the receptor structure is not known and when the ligand structure is known. So some ligands which are going to go and hit the target, thereby exerting the desired pharmacological action. If that part of the information, if it is known, then we can take up the QSAR, quantitative structure activity relationships. So what is this quantitative structure activity relationships? Okay, so QSAR is a mathematical equation that includes all statistical methods by which the biological activities, okay, normally expressed as logarithms of important molar activities, are related with the structural elements. Okay, so as it says quantitative structure activity relationships, your normal SAR, what you have learned it, okay, what you are teaching, what we are teaching for B Pharm students and Pharm students, the normal SAR, the so and so group at the so and so position increases activity, decreases activity. That itself, rather than taking just increases, decreases like this with particular data, we can quantify it with particular numbers taken into consideration. We can rate it, we can rank them. So that kind of a process, what exactly we do it in the quantitative structure activity relationships. So in order to do this quantitative structure activity relationships, what we can do it is that we can we can do it by either by 2D QSR techniques or by the 3D QSR technique. Okay. So 2D QSR techniques is that the structure will be in two-dimensional structure. Based on that itself, physicochemical parameters are calculated, or structural elements will be taken as part of the two-dimensional structure itself, and it will be correlated with the biological activity. If it is with physicochemical parameters, then we call it as an Hans analysis. If it is with structural elements, then we describe it as an Frey-Wilson analysis. So also substituents at so and so position. So that is Frey-Wilson analysis. So the classification of QSR, it is based on the two-dimensional QSR and the three-dimensional QSR. Two-dimensional QSR, that is Hans and Frey-Wilson QSR analysis methods, and the 3D QSR is the COMFA and the COMSIA 3D QSR techniques. Okay. So now today I'm going to focus now with what time I have from here. It will be on the COMFA and the COMSIA. Okay. So what is this compound comes here? Before that, let us look into the some brief history and development behind this QSAR, and then later we'll come to the compound comes here development also. Brief history and development, you may be a little curious to know how this has been developed over a period of time. So now it has been for the last 40 years since the quantitative structural and total relationships which have come to the forefront of this okay, industry. So it has been available now in agrochemistry, pharmaceutical chemistry, toxicology, and events of most of the events of the medicinal chemistry and chemistry take it into consideration. But if you look back to the history-wise, take into consideration, Crumb, Brown, and Fraser in the year 1868 itself, they have expressed an idea of that the physiological action of the substance is in a certain biological system was function of its chemical composition. In a sense, okay. So this phi is equal to function of chemical composition. The biological activity is a function of its chemical constitution. Okay. So this is what is even the QSAR what we do it now. Okay, the 2D QSAR or 3D QSAR, it was been given in the 19th century itself by the Combinatorial and the Fraser in a very simple way. Okay, so thus an alteration in the chemical constitution. Okay, we can alter this C and make it C with alterations. Okay, there will be alterations in the biological activity. So this was been given in a nutshell, like how Einstein gives theory of relativity. So this is the equation which has been given in the 19th century itself at the advanced time. Then it takes nearly 1 century later for Hans to come up and say this QSAR, quantitative structural relationships. Then the QSAR terms comes in the picture. Okay, everyone gets fascinated with this QSAR line, and everyone starts wondering about QSAR line. Okay, so this first application of QSAR is attributed to the Hans. You can see the Hans in this picture. Okay, so in the year 1960s, he starts working on that, and finally in the year 1969, he comes out with the equation, for example, like this: log of 1 by C. Where C is the representation of minimum effective dose. So it is a function of its physicochemical parameters. The physicochemical parameters are, for example, log P, which is a partition coefficient, and sigma, it is an Hammett substrate concentration. It is an as you know, it is an electronic parameter, electron donating, electron withdrawing effects based on the sigma parameter, and then the electrostatic parameter, that is tough steric parameters take into the consideration, the steric parameters. So now you can see that K1, K2, K3, K4, they are the regression coefficients, okay, constant derived from the regression analysis. Okay, so by doing the statistics, that means what they have done is they have taken set of molecules, and their molecules, the biological activity is taken as a dependent variable, and then they have done the linear, multilinear regression analysis. Okay, with if it is only one parameter, then it is a linear regression analysis. If it is in two different parameters or more than two, then it is a multilinear regression analysis. So here it is log p, sigma, and a state of steering parameters have been taken into consideration, and then they have done the regression analysis statistics to identify the 2D QSAR equation for that particular model, take into the consideration like that. Okay, so now this is what we can understand from the 2D QSAR, what we get it. That means finally we optimize the model and we come out with a better model. That means we define what is K1, K2, K3. That indicates that how much of that particular parameter should be there in that particular case, in that particular set of molecules to exhibit optimal biological activity. So that it indicates. So one, by looking at the structure, if we calculate the physicochemical parameters, okay, using computer or by manually, and if you fit into that statistical equation and if you calculate, okay, you can always predict that how much is the activity of that particular drug structure, what, yeah, okay, like that. So even before synthesis also, but 2D QSR itself we can able to design it. Okay, dear students or my dear colleagues, don't be under the impression that for doing the design you need to have very sophisticated computers or sophisticated um, um, softwares to design the drugs. Some simple techniques also you can adapt and you can design the drugs. Just okay. So one such techniques is like this: 2D techniques like that. Okay, like hands techniques, free Wilson technique. Okay, free Wilson technique is even simpler. Okay, you can always design the drugs like that. But today's my topic is focused on the three-dimensional QSR methods. That means that since we have computers now, so very advanced computers are coming up with increased RAM and okay, smart data. We can handle large number of data. We can handle it with the systems what we have today. So definitely we can do three-dimensional QSR. Okay, like that. So in this context, what happens is that important regions of the bioactive molecules are mapped in the three-dimensional space. Okay, if you think that this is the this is the molecule, this is the ligand, it is mapped surrounding the this molecule like this. So now you got some colors, you got, okay, so it can be mapped. Okay, so and it in the three-dimensional space, and we can add the fields surrounding that three-dimensional fields. Like for example, we can add hydrophobicity with one color, electrostatic parameters, steric parameters, okay, hydrogen bond donors, hydrogen bond acceptors, like that. We can few different fields we can apply and render them to overlap, and then we can develop the 3D pattern of the functional significance regions of the drug we can determine. Okay, so now as part of the 3D QSAR, when we do, what is the difference between 2D QSR and the 3D QSR? The main difference is the 2D QSR we use two-dimensional structure of the ligands, okay, or the molecules. In the 3D structure, in the 3D QSR, we use three-dimensional structure of the molecules. Okay, but provided if your prediction of the ligand's three-dimensional structure, if it is correct, then only your model is correct. If your three-dimensional structure is wrong, that means the modeling when you do it to simulate that ligand in the computer screen for your analysis as part of the QSR analysis. If that three-dimensional structure is wrong, your analysis also goes wrong. As simple as that. So your structure should be correct. Three-dimensional structure should be correct. Then only our model will be all right. So in this context, what happens is we come across like COMFA and COMSIA. Okay, what is mean by COMFA? COMFA is nothing but comparative molecular field analysis. Okay, so whereas COMSIA is nothing but comparative molecular similarity indices analysis. Okay, so for the first time in the year 1980, Kramer, a scientist from United States of America, he comes forward and he comes forward to explain about comparative molecular field analysis. So you can find one article in JACS in the year 1980 by the Kramer. It is very famous article. We we cite this article whenever we report this three-dimensional QSA. So now what Kramer introduced this compound COMSIA in the year 1980. What exactly he did is he took a set of chemical related compounds with common substructure. He takes it, okay, and he sketched and minimized their energies to get the three-dimensional structures. Once what we do is we try to sketch the ligands. Before that, what we have to do is even in the process of any 2D QSA or 3D QSA, what we do is we select bunch of molecules, and in that bunch of molecules or the set of molecules, we divide them into training set and the test set. Like okay, so what is mean by training set? Training set consists of molecules which consists of more number of molecules. Test set consists of less number of molecules. Okay, as the name implies, like test set means, for example, why we take less number of molecules in the test set is that, see, if you are any rice, if you are cooking, if the rice is boiled or not, in order to know that, we can, if you take one or two, okay, we can just check whether rice is boiled or not, just like that. So in the test set we take very few compounds, but in the training set we take more number of compounds, like okay. So set of chemically related compounds with the common structures substructures are required for both the training set as well as for the test set, like that. And the training set should have a diversity of the structures as well as the biological activity variation also should be there. Okay, that means low activity, medium activity, higher activity, that kind of diversity should be there. And so like that, next what we do, all these molecules, the training set molecules and test set molecules, in a different database we try to sketch them. Okay, when we try to sketch them, it is a two-dimensional structure only, and once after the sketching that molecules, we need to minimize their energies, we need to minimize the energies. So when we, that is what molecular modeling is all about, to convert the two-dimensional structure to the three-dimensional structure. Two-dimensional structure is not the way how it exists in the nature. Three-dimensional structure is the way how it exists in the nature. So it needs to be, two-dimensional structure needs to be converted to the three-dimensional structure. So there we use the principles of quantum mechanics and molecular mechanics. So quantum mechanics precisely for 50 atoms or less than that, if it is there, then we use quantum mechanics. If it is more than that, then we use molecular mechanics. So when we use this one, we need to optimize the bond lengths, bond angles with the two-dimensional structures, torsion angles. Okay, um, so all these things, an angle between the planes, and everything needs to be optimized, then only the three-dimensional object will be seen, just like how it exists in the three-dimensional structure like this. Okay, it is simple methane. So methane has got a bond length of 1.09 angstroms between carbon and the hydrogen, and then 109 degree 28 minutes between the angle between these bonds. So just like that, this is like a regular tetrahedron like, okay, structure like this. So all these the way how it exists in this nature, like that it needs to be simulated. Okay, so next, so what we do is we do the conformational analysis to identify the right conformation. Okay, like for example, if you take encyclohexane, it exists like the core form, chair form, but the chair form is more stable. It tends to exist in the chair form, okay, naturally in this environment. Okay, so what happens is we need to identify the right conformation, and then we have to put it in the grid box, which is from 2 to 4 angstroms size grid box. We'll put it, and then we try to before putting into the grid box, we have to add charges also, because these atoms will carry charges. Just like in periodic table of elements, so different elements, their electronegativity, electropositivity will be different. So some are electronegative, some are electropositive. So we have to give the right charges for all these atoms. Okay, then we can name that molecule and we can put it into the training set, test set. Okay, then what we have to do is we have to align these molecules. There is an important step, that is they have been aligned in the grid. So in that grid, a grid is nothing but like a cube. Okay, in that cube it is being aligned. Suppose if you are sitting in a room, we just assume that molecules are placed in that room, okay, with an x-axis and the y-axis with respect to the experimental activities and the predicted activities. So the COMFA fields needs to be applied now with for these molecules in the grid. So COMFA fields are nothing but only two. What are these COMFA fields? One is steric field and another one is electrostatic field. So these are the fields which will be applied. Remember, COMFA means very simplest one, which came first in the year 1980. Then after 10 years later, COMSIA comes in with the additional three fields. Okay, so first in the COMFA which has come, it is even famous today in the field of drug design. We use it. That is, in the COMFA they use only two fields, one is steric field and another one is electrostatic field. Only two fields have been used. Okay, so now in the COMFA align, once we align these molecules, you can see that in this structure they get aligned. Once you get aligned, you can find out the pharmacophore features connecting these pharmacophore triangles. Between the different structural components we get different triangles. Okay, so we can establish the pharmacophore once you do the alignment. Why do we do the alignment? First of all, because these molecules are going to bind to the one particular receptor, so they need to be aligned to rank them later for their activity. So that after doing that, after applying these uh, fields like a steric field and electrostatic field in the case of a compound. Okay, so steric is a physical field and electrostatic is electronic parameter. We are going to apply it. It is a three-dimensional parameter. Within these fields, there will be different components will be there. So as a variables, if there are more than three variables are there, okay, so then there will be a high dimensionality of the data will be there. So that is why we use an principal component analysis to reduce the dimensionality of the data what it is there. So principal component analysis extracts the most important factors as a principal component from the larger data it is there. Okay, so when we apply these COMFA, COMSIA, COMFA columns, okay, in the case of COMFA it is steric and electrostatic field. In the case of COMSIA, apart from steric and electrostatic field, hydrogen bond donor, hydrogen bond acceptor and hydrophobicity is also been added in. Okay, so hydrophobicity is also linked to the partition coefficient, which also plays a vital role, very important role. So all these five factors will be taken into consideration. So variables which explain the most of the data variation has been seen in this. Okay, so they have been, when they have been applied within that, there will be different components. So it will, we have to handle that certain that data means, so against the three-dimensional structure of the molecule when we apply fields. Okay, so there will be lot many components associated with that. So lot many variables associated with that. So in order to handle that, we have to use some sophisticated statistical technique like PCA or PLS. So as part of the PCA, we can see that principal components describe the variance in the independent variables, that is the descriptors if we take into consideration. Whereas in the case of PLS, the principal component describe the variance in the both the independent variables as well as in the dependent variables it shows. Okay, that is the dependent variable is the activity, independent variable is the descriptors, that means your fields take into consideration. Okay, so definitely PLS is a better version, so that is what has been adopted in the software package called as Sibyl. Okay, so Sibyl comes, it was been developed and implemented with the Conf and COMSIA. With that, the Sibyl software will becomes very famous. Sibyl software package. So you can see that partial equations based on the PCA. Now you can see that how the equations are developed for the each and every compound in the training set, how it develops it. Okay, so just learn like whereas here x is an independent variable, y is the dependent variable, take into the consideration, and all these equations take into the consideration. Now how do we, what do we get by doing this? Okay, once you apply the statistics now, we can identify the right model of that particular model what we have created, what the 3D QSR we have done. We need to optimize and get an right model. Once you get a right model, that I'll tell you in the next case study. One case study I'll take and explain with the different parameters that we take into consideration in 3D QSR next. Now finally what we get is a contour map will be getting it. So in this contour map we can see that finally, so that is yellow contour is there where the region of steric extension will not favor it, where there is a green, the steric extension will favor it. That means where there is a green counter, if you extend it further, that structure, it will increase the activity of that ligand. So where there is an yellow, there we should not increase the bulkness or steric extension should not be done like that. So like that, if you take a blue contour, where there is a blue contour, okay, electronegativity will favor. Where there is a red contour, the electropositivity will favor like that. So this is with respect to the comfort take into the consideration. If it is a COMSIA, if you take into consideration, so then hydrophobicity also comes in. We have to give different color code, and hydrogen bond donor, hydrogen bond acceptor also comes. We have to give the different color codes for that. And now in this 3D QSR, what is the beauty of this 3D QSR is that we'll get a contour map like this. It guides us to develop the newer molecules. That means newer molecule, here you should not do steric extension. Here you should do the steric extension. This side you should not do. The other side you have to do the steric extension. Here you have to keep the electronegativity groups like nitrogen, oxygen, fluorine, like that. So here there should be electropositive groups like hydrogen, for example. Okay. So that is what we do it like that. This is what is the indication how we interpret it. Now, for example, one particular case study, if I take into consideration, the COMSIA study we did it, for example, I am telling you, to make you understand what is this COMSIA is about. So, comparative molecular similarity indices analysis is COMSIA is all about. Okay. What we did is evaluation of aligned 39 compounds in the training set of taken into consideration, and so to establish the various models we have in this. So 39 compounds we have taken it in the train compounds. Okay, and the 11 compounds in the test set compounds we have taken. Totally for 50 number of compounds we did the COMSIA analysis. Okay, this the these ligands for lidosomes. Normally lidosomes are used for anti-diabetic activity. Okay, to lower the blood sugar level. That means in order to reduce the insulin resistance. And okay, so the rest of the compound constituted the test to validate the this compound COMSIA studies. So what we have used is Civil 6.7 installed on Silicon Graphics Workstation. IRIX 6.5, it is a older version, we have used it. So this work, now you can see that once the molecules are aligned, this is how the molecules will be like this. So once these molecules, when you align it in a cube, okay, so you can see that these yellow dots here, where you can see that the yellow dots are inclined with respect to the cube where we have experimental activity and the predicted activity. Okay, and after aligning, once we do the statistical analysis, okay, so we are going to predict the biological activity of these molecules and then we are going to align it. So these are all the statistical parameters that we take into the consideration. What are all the statistical parameters that we look into? Okay, the one is for example q square value is nothing but cross validated correlation coefficient, and then R square value is correlation coefficient itself. Normally the R square value will be much bigger than the cross validated Q square value. This, okay. Next is standard error of estimate. It should be least as it can be possible, and Fisher statistical significance as much as bigger as it can be possible. And we do the this uh, the cross validated R square value by bootstrapping method. We can bootstrap it and enhance the cross validated this correlation coefficient. So you can see that, for example, if the correlation coefficient is there 0.776, by bootstrapping it is enhanced to 0.811, like that. Okay. So next is number of components that has been taken. When you do this analysis, automatically it will gives the principal how many number of components we are going to take into the consideration. So which is the optimal number of components also, it will predict and it will gives us the optimal number of components line. Next is p-value, statistical significance, as you all know that it should be 0. Okay, so then only that statistical significance will be there for the. Next is these are the 5 parameters, that is steric, electrostatic, hydrophobic, hydrogen bond donor, hydrogen bond acceptor. These are the 5 parameters that we take into the consideration. So okay, so their relative strengths, you can see it. When it is only steric and electrostatic, it is there. So it is altogether this value corresponds to 1. 0.385 plus 0.615, if we add together it becomes 1. So like that, when it is only 3 fields are there, this is this much, like that. Okay, so when it is these 3 fields are there, this the contributions of these individual fields, steric, electrostatic, and in this case hydrogen bond donor to the model, if you take into consideration. Okay, based on this only you are getting this correlation coefficients. Okay, so next is this cross-validated q-square can also be done by leave one out method. Okay, usually it will be done like this. In this case, it is done by leave one out method. Like that. So next, you can see that using steric and electrostatic fields and steric hydrophobicity and electrostatic fields and steric electrostatic fields and donor, steric electrostatic and acceptor, steric electrostatic donor and acceptor. Okay, so steric, electrostatic, hydrophobicity, and donor, all the different combinations we have taken into consideration. But you can see that when all these 5 are taken together, you can see that we are finding better R square value, we are finding better the Q square value also, better the bootstrapped correlation coefficient also, we are finding it better. So that is why we see that this all the 5 parameters together, it is a better model, like that. And we say that this is a better model. So amongst all these models, okay, we have selected this model based on these figures. You can see that. Okay, So this is the best model for this 3D QSA like that. So now you can see that the once the model is been identified, then we can predict the test set compounds, and for that predicted test set compounds also we can find out the R square value for the predicted test set. Okay, that means if you have some 5 compounds, in this case 11 compounds are there in the test set, if you predict their activity, and that if you put it a regression line, if you plot it, and if you find out the correlation coefficient of that predicted and the actual, if you take into consideration, so that is the predictive R square test for that set. So that is you can see that it is better for this model as such. If you take into consideration, it is very predictive like that. It was not in the earlier cases like that. So based on this, we say that this model is a better model. So this model is finally ultimately we are going to report it in the context of other models take into the consideration. Like, okay, so now what happens is we'll get in contour maps. This is very important. Till here it is just a click of a buttons in the okay in your system, but once after here, once you come out to the contour maps now, in the contour maps you need to interpret it. That means you know that which will favors, which color code will favors the activity, which will not favors the activity with respect to these fields. So that you need to interpret now. So your role comes into the picture here. So you should actively you should take an interest, and you have to, with your patients, you have to dissect this contour map, and you have to write the structure activity relationships line by line. You have to draw the structure activity relationships for your set of and molecules what you have done it. This conf or comp, what exactly you have done it. So this is how we write the structure activity relationships. When we write from the aligned set of molecules, we get the pharmacophore features, and then the other steric, electronic with respect to hydrogen donors, acceptors, as well as hydrophobicity, where it is enhanced, where it is not enhanced, which part of the structure. You should know about the structure, chemistry of the molecules. Based on that, you have to say which will enhance, which will not enhance, like that. Based on that, you have to make it these structure activity relationships list. You have to make it like that. Okay, so this is what my dear colleagues, what I feel is this is the testing. Most of the students or my own colleagues, if I see, they can do some clicks and all, they do it once they get data. If I ask them to sit and do the interpretation of that, discussion of that, then they fail to do it, because it takes a lot of your patience and involvement. So here you have to really your role will comes into the picture like that. So altogether, what is my take home message is that COMSIA or COMFA is a very good robust ligand based drug design techniques, even though they are decades old. Okay, but still they find decay in the drug discovery and drug design in the field of drug discovery. Okay, still they are promising, I want to say. Okay, so they are basically on 3D QSAR based studies. 3D QSAR, so that means if you take see, this is the 3D QSAR. This is like, because three-dimensional structure of the molecule we are taken into consideration here, and we applied the fields and we are comparing it here, and this is how it looks like that. Okay, so now very valuable tool for the field optimization. So, so what we can see, it is can be used to predict the activity of the newer compounds before their synthesis. Okay, so that means that any new compound in this COMFA or COMSIA model, if you have developed any new compound, you bring it in this contour map, and you can predict their activity. Okay. In this developed contour model, what we have developed, the three-dimensional 3D QSAR model, you can bring in any molecule inside that, and you can predict its activity like that. in that control maps, in that changed model. But the synthesis, the question is that you can predict any molecule before its synthesis, it can be predicted in the contour maps. But the question is that conditions apply in the sense. Just like how you see the advertisement with the conditions with the star marks like that. So conditions, what is the conditions is that see this ligands, the ligand should contain the same scaffold as that of the ligands which are already there in the training set of that model development. Okay, so then only that predictions will be quite accurate. Otherwise, if the common scaffold, if it is not there, that will not be accurate. And also, if you have more number of compounds in the training set, better the prediction of the newer compounds activity, like, and more the diversity of the structures and diversity of the activities, it is more the prediction of the activity like that.